Hello and welcome back. Today we are going to dive into really Punnett squares, where we're going to talk a little bit about Mendel again. We've talked about him briefly, but we're going to really focus in on the laws that he made that kind of showed what genetics is, how inheritance works, and laws that we still use today. So let's get started. Just right off the bat, I want to do a quick review of who Gregor Mendel is and kind of the basic vocab again. So Gregor Mendel is considered the father of genetics. He studied pea plants, like that was his big thing, it was pea plants, and he studied them by looking at one single trait at a time. So he only focused in on one thing, and what we call that is a monohybrid cross. So mono means one, hybrid means mixing, so you're crossing really, you're mixing only one trait. That's what monohybrid means. Now, we're going to use these following terms when we're talking about Punnett squares. So when we're talking about the parents, we call them the P1 generation, P standing for parent. And then when the parent generation, the P1 generation, make offspring, those offspring are called the F1 generation. The F stands for filial. It's like an old school term that meant children. We don't use that term anymore, but the F of that word did stick around. So we call the first offspring produced F1. And then when you cross offspring from the F1 generation to make new offspring, these would be like the grandchildren from the parent cross. Those are called F2 generation. It sort of makes sense, right? P1, P is parent, and then F1 is the first offspring, F2 is the second generation of offspring. And then just as a reminder, all traits that we're going to look at have two alleles of that trait, so two versions of that trait, right? So you could have like tall plants or short plants, green seeds or yellow seeds, only two versions. Now the title of this set of notes is called Punnett squares, and so we're going to focus on using Punnett squares to figure out potential probabilities. Now that's literally the point of a Punnett square. The purpose is to predict the probability of traits appearing in the offspring. Now when we do a Punnett square, it doesn't necessarily mean that whatever we put in the Punnett square is like absolutely has to happen. This is like, I'm laying down the law, like my Punnett square says this. No, that's not what it means. A Punnett square just means that this could happen. This is the probability that it will happen, but nothing is 100% guaranteed just by doing a Punnett square. So the way that we set up a Punnett square is on the outside of our little like window shape here, on the outside is our P1 generation, right? So we have the father, whoever the father is. It could be just like one plant that we're like dubbing the father here, okay? We have father's genotypes on the top, mother's genotypes on the side, and this is just their alleles that they have that they're going to be crossing. And then you put the offspring in the actual window shape. So the first thing we're going to do is we're going to cross and like put together these two A's from that would intersect in this very first window, and that will give us a homozygous dominant genotype. And you just sort of continue on. You're just crossing the genotypes, and wherever they would intersect on the Punnett square, you're just filling in whatever that is. So our second genotype is a heterozygous genotype, and as you continue on, you'll see that another homozygous dominant and another heterozygous. And all you are doing is literally just wherever these alleles are going to intersect with each other on the Punnett square, you just write it down. That's pretty much it. So let's get into our rules, our steps that we're going to follow, and then we're going to do one practice Punnett square together. So our rules here, there are 5 steps. Number 1, make a key for the alleles. Remember that if you are looking at a trait, you use the same letter for the two versions, except the capital letter notates dominant and the lowercase letter notates recessive. So even though we're going to use the letter R here, right? Round starts with R, right? That makes sense. Wrinkle doesn't start with R, but we're still using the same letter. We do not use two different letters. Get that thought right out of your head right now. You use the same letter. Step 2 is to write down the parent genotypes. This will typically be given to you in a word problem, so you're just like writing it down so that you have it so that you can reflect back on. Then you draw the Punnett square, you add the P1 alleles on the outside, and you find the F1 generation. And then step 4 and 5 are just determining the ratios, genotypically and phenotypically, of the F1 generation. Now, this is a lot of words, but let's put this into practice. So, this would be an example problem that you might get on like a test or something. I'm telling you that black fur is dominant over white fur in guinea pigs. And I want to cross 2 heterozygous black fur guinea pigs. Okay, so step 1 is figuring out our allele key. So I was told that black fur is dominant, so I'm going to give black fur that capital B, and I'm going to use B because black starts with B, and I don't know, that's just easy for me to remember. You can use any letter, but I'm just sticking with ones that make sense to me. And then white fur is recessive, and so I'm going to give that the lowercase letter. Step 2 is to determine my P1 genotypes, right? My parental genotypes. I know I have 2 heterozygous guinea pigs, which means I'm going to have 2 different alleles for mom and 2 different alleles for dad. And so, there we go, right? A capital B and a lowercase b for mom and a capital B and a lowercase b for dad. My third step is to determine my actual Punnett square. So I just set that up like I have set up the one on the previous slide, right? I have my first genotype on the top, my second genotype on the side in these like grayed out boxes. These are my P1 alleles. And then again, I'm just, wherever these alleles intersect, I'm just writing what the 2 alleles would be. So in this first box, I have a capital B and a capital B. They're going to intersect together, and that's exactly what I'm going to write. In this second box, I have the capital B from one parent and the lowercase b from the other parent, and so I have a heterozygous genotype. In the third box, I have the lowercase b from one parent and the capital B from the other. Now, you may have been tempted to put the little b before the big B just because you were putting all of, the left side genotype first, the letters first. Don't do that. Always, always, always put the capital letter first when you're filling this out. It just makes it way easier to read in the long run. And then our last one, we have 2 lowercase b's that are going to intersect, so there we have it. For step 4 and 5, we are strictly looking at the offspring, so only the white boxes, and we're going to write out our genotypic ratio. And all that is like how many big B's do you have, how many big B little b's do you have, and how many little b's do you have? Well, I have 1 big B, 2 big B little b's, and 1 little b. And that's all I write. That's my ratio, right? 1 to 2 to 1. And then for my phenotypic ratio, I have to look at what these genotypes mean. So how many of these are going to show up as black fur, and how many are going to show up as white fur? That's what the phenotypic ratio is asking. And in this case, I have 3 black fur and 1 white fur. Any offspring that has a capital B is going to be showing as black fur, and then the one genotype that has little b will be white fur. Okay, we're going to practice this a lot more, but we're not quite done yet because it does get a little bit more complicated. So let's talk about Mendel again, my buddy Mendel, who came up with the laws. There are really 3 that I need you to focus on. Well, there are only 3, but these are the ones that we're going to talk about in class. So law number 1 is the law of segregation. And what that means is that when gametes are produced during meiosis, the alleles separate. So gametes only have 1 allele for each trait. That makes sense, right? You only have 1 copy of every gene in an allele, right? They're haploid, they have half the number of chromosomes, so 1 allele. The law of independent assortment is just saying that alleles get inherited independently of each other, meaning that just because you get one allele doesn't guarantee that you get a second allele, right? You could get brown hair and brown eyes, but it doesn't guarantee just because you have brown hair that you'll have brown eyes, okay? They get inherited separately from each other. And like in cats, tail length and hair color are not inherited together. They get inherited separately or independently from each other. And then the last law is the law of dominance, and this is exactly what it sounds like. One trait is dominant over the other. This may sound at this point like, yeah, duh, I knew that, law of dominance, yeah. Yeah, well, this was pretty groundbreaking stuff back in the 1700s, you know, so yeah, there's a law about it. So that one is like probably the easiest one to remember, but the other two are the ones that we are going to focus on the next slide. So we talked about Punnett squares, and every Punnett square that we did was a monohybrid cross, right? We only crossed one trait. Black fur or white fur, that's still just fur color. The trait itself was only one. But you can study two at the same time. You can actually study more than that. You can do 3, 4. It gets crazy, but we're going to probably stop at 2. But an example of this would be in pea plants. You can study both plant height and seed color at the same time. And what this really just looks like is you break it down, but you just have more letters in your genotype. So a plant that is heterozygous for plant height and homozygous recessive for color would just have the heterozygous genotype for the height, right? Capital T, lowercase t, and then homozygous recessive for color, so 2 little g's. We put them together, so it's 4 letters in a row, but really you're just kind of breaking it down per trait and then smushing them together at the end. Another example of this would be a pure tall plant. Pure means homozygous, so 2 of the same, and tall would be the capital T's. And hybrid, green seeds, hybrid means a mix, so that would be like heterozygous. And so you would have capital T, and then capital G, lowercase g. So again, you are figuring out the genotypes for each trait and then just smushing the letters together at the end so that you have 4 in a row instead of 2. And this is our last slide, but when you have a dihybrid cross, remember that the law of segregation stated that you can only receive 1 allele of each trait. So when we are figuring out what our gametes are, we actually have to figure out how those alleles could be inherited into those gametes. So using that same key from the previous slide, we're going to figure out this genotype's possible gametes, okay? So I'm telling you that I have a plant that is heterozygous for both height and for color. So big T, big G, little g. Okay, that's my parent. I've got it right here. I told you that's the parent. And then I wrote it bigger over here so that we can figure out what our gametes are. Now the best way to do this is the FOIL method, and maybe you've done this in math at some point in your life, but I'm gonna refer to it as the FOIL method, and that's what you're gonna see on the screen as well. So for the FOIL method, you start with the first letters, and when I say first, I mean the first letters of each letter type. Okay, so the first letters would be the first T and the first G. Okay, that's one possible gamete. Once you have the first, you go to the outside letters of this genotype, and for that it would be the big T and the little g. So I have my first letters, my outside letters, then I'm going to do my inside letters, and then I'm going to do the last letters of both letter types. So the last letters of the T's and the last letters of the G's to give me 4 possible gametes. And you can see that I did the FOIL method, right? First, outside, inside, last, FOIL method. Now my possible gametes, once I pull all that information out, would be capital T, capital G first, capital T, lowercase g outside, little t, big G inside, and little t, little g last. I told you before that you should always put your capital letters first. But in this case, when you do dihybrid, you just want to always have your T's first, in this case, because we're using T and G. So regardless, the T is always going to come first, even if it's lowercase and the G is capital. It's just like little tricks here that are going to help you when you have to figure out like genotype and phenotype ratios. But we're going to practice this way more in class because this is and can be kind of confusing. But once we kind of get into doing the Punnett squares, I think you're going to have a lot of fun. I really like this unit, and I hope that you do too. All right, I know this was a bit long, but great job, and I'm super proud of you.